In lab, we're going to run an exercise using something called restriction enzymes, and it turns out that restriction enzymes are tools that you'd expect to find in basically any molecular biology lab that's running out there. Basically what restriction enzymes do, like this picture shows, is that they're enzymes that cut DNA somewhere along its sugar phosphate backbone. And so we'll see that restriction enzymes actually have a few different uses by molecular biologists. So molecular biologists use restriction enzymes for a few different things. Where restriction enzymes are naturally found, though, are in bacteria. So it turns out that, you may or may not know this, but bacteria can be infected with viruses. And so kind of as a way of protecting themselves against viral infection, what a lot of bacteria can do is that they can make these restriction enzymes. And the purpose of making them is to chop up any viral DNA that tries to infect the bacterial cell. So it's kind of a rudimentary defense mechanism that bacteria have against viruses that try to infect them. And so again, restriction enzymes, all they do is that they're enzymes that cut DNA. It turns out that restriction enzymes don't cut DNA just in any old place. So a couple of things to know about restriction enzymes is that they cut DNA, but at specific spots. Right, so we'll see that there's actually different types of restriction enzymes out there. And the way that these different restriction enzymes differ from each other is the fact that, one, each type of restriction enzyme will recognize a different sequence on DNA. Not only that, but each restriction enzyme will cut the DNA in a different way after it recognizes the sequence that it's specific for. So to kind of make sense of all of this, to see how restriction enzymes work and what they do, how about if we take a look at a couple different examples of restriction enzymes and what they do. One example of a restriction enzyme is a restriction enzyme called EcoR5. And so again, EcoR5 is a restriction enzyme, which means that what it does is that it cuts DNA somewhere along the sugar phosphate backbone. It can only make that cut, though, if a certain sequence is present within the DNA. And it turns out that this restriction enzyme, this particular one, EcoR5, will only cut DNA if it sees this specific sequence, GAT-ATC, and then here's its complement down below. And so if EcoR5 happens to run across this specific sequence, then and only then will it cut within that sequence somewhere. And the way that EcoR5 cuts is that it actually makes a cut between the T and the A, and then the T and the A down here. And so if you were to apply that to an entire DNA strand, so say that we have, here's a portion of a DNA strand, and if we treated this DNA strand with this restriction enzyme, EcoR1, what we're saying is that EcoR1 will cut this DNA in half only if this particular sequence is present within this DNA strand. And so if you kind of scan through this DNA strand, you'll see that this sequence, it indeed appears within this DNA strand once. And if you haven't spotted it, here it is. So GAT-ATC, that matches this, GAT-ATC. And what we're saying is that because this specific sequence is present within this entire DNA strand, we're saying that EcoR1 will be able to cut this DNA strand, and where it will cut it is within this sequence, specifically between the T and the A, and the A and the T. So that's exactly where within the sugar phosphate backbone that EcoR5 will end up cutting this particular DNA strand. And after it cuts it, after it makes these little nicks in the sugar phosphate backbone, here's what results. So after that sugar phosphate backbone is cut, you'll end up with now two pieces of DNA. Right? So here's one piece or one fragment of DNA, and here's another piece or another fragment of DNA. So when EcoR5 was used on that original piece of DNA, what resulted was one cut. And because it was cut once, it resulted in two pieces of DNA. Again, this one over here and this one over here. One of the things that molecular biologists are interested in often is how big the pieces are that resulted from a restriction enzyme cut. And so there's different ways of describing length, but conventionally the way that DNA length is described is in terms of how many base pairs it contains. So a base pair is, for example, this GC. That's one pair of bases, or one base pair. GC is another pair of bases, or another base pair. AT, that's another base pair. So again, what we're saying is that DNA fragments are conventionally described when you want to talk about its length in terms of how many base pairs it has. So if you want to describe how big this fragment is, you just count the number of base pairs. Same thing here. If you want to describe the number of, or if you want to describe how long this DNA fragment is, then you'd count the number of base pairs that it contains. And what we've just done is highlighted the base pairs to make it easier to count. So if you just count those turquoise highlighted base pairs, you'd see that there are 6 base pairs within this fragment, and there are 11 base pairs within this fragment. So in other words, this fragment here, it's 6 base pairs long. That's how its length would be described. This DNA fragment is 11 base pairs long. Typically, if you're talking about a real-life DNA strand, like real DNA strands are really, really, really long, like millions of base pairs long. And so typically what is done is that base pairs aren't described in terms of, or length is not described in terms of base pairs, but instead in terms of kilobase pairs. So just a useful little conversion factor here. Whenever someone is talking about 1 kilobase pair, that's equal to 1,000 base pairs. And again, conventionally kilobase pairs are used when describing DNA length because DNA, real DNA, is made up of millions of base pairs. So again, what restriction enzymes do is that they cut DNA, but only if they see the sequence that they're specific for. It turns out that not all restriction enzymes cut like EcoR5 cuts. And so here's what we mean by that. Another example of a restriction enzyme that's out there is something called TACA1. And what we're saying is that TACA1, it's different than EcoR5 in terms of not only the sequence that it recognizes, but the way that it cuts. And we're saying here that this enzyme here, TACA1, just TACA1, makes these things called staggered cuts. Here's what that means. What the specific sequence that TACA1 recognizes is this one. So what we're saying is that TACA1 will cut DNA only if this specific sequence is present within the DNA. If TACA1 sees this sequence, it'll cut within the sequence, but it'll cut it in a staggered fashion. And here's what we mean by that. It turns out that TACA1 will cut between the T and the C here, and the C and the T down here. So it's not a straight through cut like we saw EcoR5 making. Instead, it's a staggered cut. And so the thing that results from a staggered cut is this. So say that you take a piece of DNA and you treat it with TACA1. TACA1 will only cut this DNA if this specific sequence, TCGA, is present within this DNA. And if you scan through this DNA, you might see that this specific sequence is present, and it's present once within this DNA. And there it is, TCGA and the complement AGCT down below. Because this sequence is present within this DNA strand, it means that TACA1 will be able to cut this DNA strand. And where it will cut it is within the sequence that it recognizes, specifically between the T and the C here, and between the C and the T down here. And so if nicks are made right here within the sugar phosphate backbone, and here within the sugar phosphate backbone, check out what results. 2 DNA fragments, but the DNA fragments that are produced kind of have these overhanging single-stranded portions. And what these overhanging single-stranded portions are called, they're called sticky ends.